So my name's Tom Denton. I've been at Google for 10 years and taught at York 10 years and a bit ago. And I lead a small team of folks working on bioacoustics at Google. And the name of that project is Perch. And let's do slideshow mode. And that looks like maybe it looks good. Maybe give me a thumbs up, Eric, if you can see it. Great, cool. So our mission as a team is to organize the sounds of the natural world and make them universally accessible and useful. And we work with groups doing bioacoustic monitoring really around the world. I think we've done projects with people on just about every continent but Antarctica. And I've got a friend that goes to Antarctica to listen to whales, so I think we might fix that soon. So why bioacoustics? Let's start there. I'm going to do about maybe 10, 15 minutes of sort of context setting, and then we'll go through some of the collab notebooks that are available in our GitHub repository. So why bioacoustics? So if you imagine closing your eyes and then opening them and finding that you've been teleported to this rainforest, you might not see terribly much here in terms of animals. We've got our trees and leaves and got a lot of mist because it's humid, but because it's a rainforest, but we don't actually see the animals here. But if we were to open our ears, we might hear something like this. Were you able to hear that, Eric? No. Ha! Cool. So it turns out sound is really important for this presentation. So let's take a moment and try to figure out how to get Zoom to handle sound. Sorry, we also had some sign-in issues, so I wasn't able to test this before. How do we get this to work? Check. Let's try that again, just to see if magically it suddenly works now. Okay, so unless, Eric, if you know how to get Zoom to allow sound, I'll just start doing a search for Zoom share sound in presentation slides.
Yeah, the only thing I know is when you start sharing screen, there should be an option to share with sound.
Okay. Sure. Why don't I just try redoing this then? Maybe what I can do, let's see if this works. How about now? No. Okay. Well, Advanced sharing options. That's not what we want. Okay, well, this is really unfortunate. All right, well, maybe I'll just give you an introduction to how to read a spectrogram. Because Zoom apparently isn't good for this sort of thing. All right, so spectrograms, you have a time axis and you have a frequency axis. And in the time axis, we have time. This is 5 seconds of audio. And the frequency, we have sort of lower sounding things at the bottom and higher pitch things at the top. And in this particular spectrogram, we can see actually there's a variety of things going on. So there's these sort of like high pitch short things, which it turns out are going to be insects. There's a good bit of noise sort of in all channels. And there's quite a bit of background and sort of all of that, all of this sort of noisy stuff you can see. It's actually a pretty busy thing. And there's this sort of cackling bird sound that these sort of almost vertical things are representing. So yeah, so if we were able to listen to this, you'd hear a pretty busy scene. So you're able to hear a lot more than you're able to see in this rainforest situation. So for about 20 years or so, it's been pretty reasonable to slap a microphone on the side of a tree and turn it on and just collect a bunch of audio. So this is an example of what we call passive acoustic monitoring, and this is an autonomous recording unit, this one produced by the Cornell Lab of Ornithology. And over the years, folks have gone from putting out a few of these at a time to putting out hundreds or even thousands of them, and collecting thousands or tens of thousands of hours of audio, or even millions. Now, all of that audio, it ends up being too much for humans to listen to. And we also really need expert listeners to tell us, what are they actually hearing in this audio? So thus, machine learning is helpful. So we've got all of this messy data, and we want to try to get to the bottom of what's in there. Now, it turns out there's an enormous amount of complexity in this data. So I'll give just this one example of white-crowned sparrow. So that's one species of bird. And in these spectrograms, you can see that these are coming from across the United States. So Michigan, well, Yukon is Canada, sorry, Washington, Oregon, Missouri. And each of these sort of prominent vocalizations are different examples of white-crowned sparrow songs. And you can see that there's...
Sorry, real quick. I don't know if you're meaning to be sharing slides right now, but
yes, I am. This is a mess. Cool. All right. Thank you, Zoom, for all of the help that you are giving us today. Cool. So can we see it now? Okay, great. So, right. So these are examples of white-crowned sparrow songs. You can see that there's quite a bit of variation here. I am so disappointed that we don't have audio, because these are great. And you can see, like many of these songs, we've got this sort of opening note, maybe we've got a bit of buzziness in here, but there's also quite a bit of variation internally for these. And so the white-crowned sparrow song, we have these indicators of the species, so these opening notes, but we also have a lot of geographic and then even individual variation within those songs. We can also see that there's different kinds of vocalization. So these first 5 are all songs. So those are generally for territory defense and attracting mates. But we also have calls that are given during flight. We have juvenile calls, which often are begging calls, so asking for food, this sort of thing. And there are also alarm calls, which can be used to indicate the presence of a predator. And some species of birds have different vocalizations indicating predators in the sky versus on the ground. So you can have multiple vocalizations. Some species, there's a whole lot of different songs, not just one, with lots of different kinds of structure and functions within the environment. We see a lot of geographic and individual variation. And when we look at this one from Missouri, we can see, hey, there's actually a lot going on in there. It's not just that white-crowned sparrow. There's a lot of other birds vocalizing as well. And so we can have overlap of these songs, both in time and frequency. Now, a couple more bird facts just to keep us in the right frame of mind. So it turns out, for songbirds at least, audio time perception is much better than human time perception of sound. And so this dark-eyed junco sound, if I were able to play it back to you, thanks again, Zoom, what we would hear is something like a da-da-da-da-da-da-da-da-da-da. So it sounds like a single note repeating. But if you slow it down far enough or look at the spectrogram, you can make out these individual notes and see that it's actually this sort of alternating pairs of notes. But when it's fast enough, our ears resolve those into single notes. Likewise, there's also these special notes, polyphonic notes that birds can make that we can't. So they are basically able to have vocal folds sort of attached to each of their lungs instead of just one set of vocal folds in their esophagus. And so they can create these polyphonic notes, which are sort of like making 2 notes at the same time on top of each other. And our ears don't really pick these up terribly well. So we've got all of this detail that we actually can't get with our ears. But, if you go about learning birdsong, you can learn to reliably identify things, at least in your local area generally. And then to sort of show the overall complexity of this problem, there's 10,000 species of bird worldwide. We also have 5,000 frogs, 20,000 orthoptera, which are crickets and things like that, 1,400 bats, 400 primates, and one primate with opposable thumbs that gets probably about 99% of the attention in the machine learning space. So it's good to diversify a little bit. Now, when we come into the biodiversity monitoring problems, there's really two kinds of problems that we see, which are really at opposite ends of the scale. So you might be interested in identifying everything that's in the soundscape. That's maybe every bird or something like this. And that's what we would call a broad monitoring problem. So we want classifiers that are able to identify this broad collection of species. And in contrast, we also have a lot of problems that are specific species. So like you've got an endangered or invasive species that you want to keep tabs on. And the endangered species, you might really care, like, what is it doing in this area? Is it just looking for food, or might there be a nest nearby? Are there juveniles that we can hear, this sort of thing. And these give us a lot of information about what area is most important to protect. For invasive species, you want like early indicators because it's easier to keep them out than it is to take them out once they're in an area. And these two kinds of problems, they have very different sort of characteristics in terms of the classifiers you want to get out. So for specific species monitoring, we really want to get recall first. So we don't want to be missing any of those invasive species, and we really want to get those endangered species if they're there. And we're only looking at one or two things at a time, and so we can spend some graduate student time going through false positives to tell us whether these things are here or not. So we really want to get recall first and then try to bring up precision. On the other hand, for broad monitoring, you're often looking at so many species that any false positives are just going to, you're not going to have enough grad students to deal with it. So for broad monitoring, typically we're looking for precision first, and we might even be okay like letting go of species where we don't have high enough precision, and then increasing recall as we sort of iterate on the models. And so you can build up classifiers one by one, and sort of what we found in a few years of doing this work is this classification as a process doesn't really scale well enough. So we find ourselves retraining per use case. That sort of geographic variation is a problem. People are always coming to us with new things that they're trying to monitor for that people haven't tried to monitor for before. Whether it's some new bird species that there's almost no training data for, or specific kinds of vocalizations, or insects or frog species, or this sort of thing. So if we want to sort of train classifiers from scratch, or even fine-tune large models, the annotation process ends up being way too onerous, especially for field biologists who really have better things to do than messing around with our, silly computer science models. And what we also see is that poorer countries also tend to have a lot more species richness. So a lot more of the biodiversity is in the tropics, and relatively fewer grad students to throw at the problems. And all of these things. So, this sort of per use case development of classifiers ends up needing machine learning experts in the loop. And that ends up being really problematic. So we come at this problem and we step back and we ask this sort of big question: how do we build a system that efficiently helps us answer questions which have never been asked before? So how do we find new bioacoustic signals that people want very efficiently without necessarily needing that machine learning expert in the loop? Now, I want to point out that this really flips things around from what I often see in machine learning papers. So machine learning papers, they usually look at a problem and say like, oh, this bioacoustics problem, it's really bad because we need these bioacoustics experts. And wouldn't it be great if we could sort of factor them out of the problem? But that's actually completely upside down. They're the people that know the problems. They know what to look for, right? They know what questions are going to be useful to answer. They're the ones we want involved. The problem is the machine learning experts. So we need to get us out of the way, get our systems out of the way, and let these people run and succeed at what they're trying to do. So this is sort of the workflow of a passive acoustic monitoring project. So we've got some collection of audio data. That's our tens of thousands of hours of audio that we get from our recorders. Then maybe we want to get some training data for the problem at hand. Then we want to train some classifier. We want to evaluate it. We're going to figure out that the classifier is no good. So then we're going to maybe get more training data and train a new version of it. Then we're going to run some inference over those tens of thousands of hours of audio. And then finally, once we've done that, we can actually start answering ecology questions. And, all of these steps, they end up normally requiring quite a bit of work. So collecting the audio data, yeah, we have to put the microphones out and all of this, collect up the data, figure out where we're going to put it. And answering the ecology questions, that's largely like, let's go to R, figure out like how these inferences and detections sort of relate to the various interventions we've made and find out whether it's working or not. And all of this stuff in the middle, this is kind of the machine learning part of the problem. And the system we've put together streamlines all of these steps. So the game is going to be, we have a pre-trained model that we can use to get embeddings of our target data. And then we can search over those embeddings to find relevant examples very efficiently. So that gives us our training data in almost no time. Using pre-trained, pre-computed embeddings, we can train classifiers on top of examples very efficiently in about a minute or so. And then we can, apply active learning. So this whole loop, we can really, really simplify and boil down to its essence and get to the point where biologists can sort of click buttons in Colab notebooks and get what it is that they're after, which is a classifier for whatever this new signal is that they're interested in. And the goal is really to try to get this whole process of building a classifier for a signal to somewhere down below an hour of effort. Once we do that, then we hope that people are going to be able to step back from this and say like, oh yeah, we're going to collect our audio data, we're going to use that machine learning stuff, and then we're going to go answer some ecology questions, which is the part we actually care about. So how does the system work? Well, we're going to take some embeddings from a pre-trained model. If you haven't worked with embeddings before, this is sort of the picture of how it works. So in our case, we train a classifier. So it's a global birdsong model trained to distinguish 10,000 species of bird in sort of an all against all sort of fashion. And that takes in a waveform. We compute a spectrogram, a particularly nice one called a P-SYN-MEL spectrogram. And then at the end, we're going to pop out some probabilities for different sorts of species or things, right? So maybe we've got dove 98%, parrot 87%, koala 0% for this particular spectrogram. And then in our case, the embedding we're going to use is just the last layer before we get these, or this last set of activations before we get these probabilities. And so there's a few nice ways to think about this. You can think of it as sort of like, what is the best 1,000-dimensional vector you could ever come up with to predict which of the world's 10,000 bird species are in this 5-second chunk of audio, right? And that's exactly what we've trained the thing to do. So like that's the 1,000-ish dimensional vector that we're working with. And if that's sort of, too in the weeds or too technical, you can think of it as just a really nice fingerprint of the sound that's particularly helpful for identifying bird species. So then you say, hey, Tom, yeah, that's cool and all, but like koalas aren't birds and frogs aren't birds and all of these other things. Like, is this embedding actually good for other stuff? And the answer is yes. So we found that these global bird classifier embeddings can organize novel data effectively and transfer learn to all sorts of new problems. So this particular picture, I'll just walk through quickly. So this is a Godwit call type problem. So the first question you might ask is, hey, we've trained a species classifier. Do we lose call type information by the time we get to the end of the network? Or is the call type still sort of there for us to get a hold of? And so we compared two different bird models, so BirdNet and Perch, which is our sort of BirdNet equivalent. They're trained in very similar ways. And we compared those to a number of general audio event detection embeddings. So audio MAE, which is a large transformer trained on lots of YouTube data, YAMNet and VGG-ish, also sort of event detectors. The y-axis here is ROC-AUC, so sort of a standard evaluation metric. And the x-axis here is the number of examples, training examples per class that we used for training our linear classifier on top of the embeddings. So in this case, with just 4 examples per class, there's 5 classes in all in this problem, so 20 training examples in all, we're getting well north of 90% ROC-AUC with the bird models. And when we sort of plot out the embeddings, so this is a t-SNE embedding or t-SNE projection of the embedding. So take those 1,000-dimensional vectors, put them in 2-dimensional space, and then color them according to class. We can see that for the bird models, we're getting really nice sort of pre-clustering of these classes, which then means that they're going to be very easy to classify in the embedding space. Whereas for sort of the general audio event models, things get pretty muddy, and we sort of see that reflected in the scores. Now, it doesn't just work for call type. We also applied this to yellowhammer dialects, which is a very, very subtle difference in some different songs, so detecting geographic variation. And this one is worth talking through, yeah. So you've got a yellowhammer song. The first part has a bunch of variation in it that we don't care about. And then in the second part of the song, if it's a low note then a high note, you're in one part of Germany. If it's a high note then a low note, you're in a different part of Germany. So the question is, can we throw a bunch of examples of yellowhammer songs through this embedding process and then still be able to train a linear classifier to pull out that one note difference or difference in ordering of those 2 notes from those vocalizations? And the answer is yes, but you need a little bit more data to do it. Bats are not birds, so you might know. But if we take these sort of high-pitched echolocation vocalizations and pitch shift them down into the normally audible frequency range, we can use embedding linear classifiers trained on top of embeddings to identify different bat species. Then you might say, well, bats fly, so they're effectively birds, so that should not be easy. And it turns out, hey, okay, we can do marine mammals too. So marine mammals, we've got a little bit less of an edge there, but we've also been building some new models with a lot more underwater data available to them. But we're doing a good job there too. You say, hey, yeah, sure, whatever. Swimming is just flying underwater. Marine mammals, also birds. And then we'll get to frogs. And frogs, I would assert, are actually different from birds. And we're doing an okay job there too. And then this last one, this bird species, this is a number of birds that were not really in the training set for the original classifier. So some very low data birds from Peru, mainly. Okay, so we've got some good embeddings. We've got this sort of nice pre-clustering in the embedding space that makes these linear classification problems much easier. And it turns out that that pre-clustering also really enables search. So if we've got good embeddings, then we can take a query, so some example of something that we're interested in. We can get an embedding of that query, and then we can start matching it against embeddings in our database. So in this picture, we've got some woodpecker drumming, and these are some results from an actual search. And we're able to see, yeah, there's woodpecker drumming in all of these different spectrograms. So we're able to do this sort of similarity search and pull out examples of new things that we're interested in. Now, you might look at this and say, Tom, the previous spectrograms that we saw, they were all black and white. How do you have these really nice color spectrograms? And it turns out we've also built a really nice unsupervised source separation model, which we can use for all sorts of good stuff. And so this is the audio that we didn't hear at the beginning with the sort of cacophony of birds and insects and other sort of forest noise. And the source separation algorithm is able to pull this apart into constituent pieces. So we're able to isolate some insects here on the top. By the way, are you able to see my cursor? Yeah, okay, cool. So we've got some insects up top. Down on the bottom, we've got this sort of chattering bird. It goes, ha, 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 ha. And then in the middle, we've got this high-pitched bird, which you can see pretty faintly in the original spectrogram. And actually, most of the time when people hear this original sample, even if you listen to it 2 or 3 times, you don't really notice this until it's pulled out. Once you see it and hear it pulled out, you can hear it in the original pretty easily. So the source separation is able to do some pretty cool stuff. And it's also able to help search. So this was a small example. We were looking for glossy black cockatoos, which are a very cryptic endangered species in Eastern Australia. And this is sort of from a 2-hour audio file on the x-axis. So we've got some time here. And then this little peak, so the y-axis here, it's negative log distance. So you can just call it a similarity score. So up is good. And it turns out this little peak is a glossy black cockatoo. And these 2 peaks were not. So we've got something here. Now there's a couple of ways we can apply separation here. So we can separate the data and say, like, let's take all of our separated channels and then take the most similarity over those channels to our query. And that gives us a really, really nice peak there. The other thing we can do is we can take the query and apply separation to the query and get rid of the other background noise and the other little bird that's chittering in the background. And that gives us a nice peak too. And then if we combine those 2 techniques, we get sort of the best signal to noise ratio at all. And this glossy black cockatoo really just stands out. And so this was able to discover a population of glossy black cockatoos that wasn't previously known. So that's been really great. So that's sort of the end of the context setting piece. That only took half the time. That'll be fine. And I'll just end on that part on this quote from Karen Backer's book, The Sounds of Life, which I strongly recommend. This is kind of at the very end of the book, so spoiler alert. She says, the microscope enabled humans to see anew with both our eyes and imaginations. Digital acoustics are an invention of similar significance. Today we're hearing things we never imagined we could hear. And so we're really trying to get at all of these new things that we're able to hear with these techniques. And with that, I'll actually switch over to running through some Colab notebooks and show you how they work. I'm going to work with the notebooks that we have in our GitHub repository. And then there's a tutorial version that I think we posted for NeurIPS last year, which I think will also be shared with everyone. And you can play with those, and it has a little bit more of like the text and framing and available in it. But today I'll work with the sort of like most up-to-date version. And just to show you that we do actually work well with frogs, we're going to be working with this data set called a NeuraSet, which is a set of Brazilian frog and toad vocalizations, which covers 2 different spots in Brazil, 4 sites, 2 different biomes, and contains a total of 42 species. And we're going to work with 2 frogs today. One is the lesser tree frog, which is this cute little guy here. And the other one is the amazing waxy monkey tree frog, which is this brilliant looking beast that we see on the bottom right. So with that, I will swap over to this tab. And it looks like I've successfully connected to my kernel. Thank you. And so if you're coming into this sort of raw, a nice way to do this is you can hit this open a notebook button, and then there's a GitHub tab here. And actually so we've got our Google Perch GitHub repository. And within this, if we go over to this embed audio and copy the link address, and then come over here and back into our Colab notebook, then we can like drop that whole URL in here, and it'll be able to open that notebook directly. And so that's what I've done here. So we've just connected to that notebook. I'm running a local Colab kernel, or a local Jupyter kernel, actually. So this is just connected to a bog standard Jupyter instance where I've installed that GitHub repository as well. If you're using just sort of a off-the-shelf Colab kernel, you can run this pip install command to install all of the dependencies. If you're running your own Jupyter notebook, you can install that locally, install the Perch repository, and then get that started with those dependencies already in place. So because I'm running on my local one, I've already got the dependencies installed, and so I can skip this installation step. So I'll run this thing, get a warning that even though I wrote it, it was not authored by Google. And then we're going to set some basic configuration. So we have a number of models we can work with. We've actually sort of set up the whole thing so that you can pick up, you can use our model, which is Perch. It's a good model. I've worked hard on it. And let's say we're going to call this a neuroset demo. So we're going to put all the stuff that we're working with in some directory. We're choosing to use Perch. You can also use BirdNet. And for other embedding models, there's a class that you can implement to use any other embedding model you want as well. So basically, just need to write some way to load up a model and then write some little wrapper method that says, here's how I take audio and spit out embeddings. And then once you do that, you can use anything you want. So actually, if you're really interested in music instead of bioacoustics, and you've got some really nice model for music embedding, you could apply all of the same stuff to it. All right, so we're going to run that. There's our configuration. And now we're going to tell it just a little bit more about our model. We're using our TensorFlow Hub version, so I don't need to tell it where the local version is. Oh, and then this other piece that's really important, we have to tell it where the audio is. So I've got some local directory full of audio for all of that Neuroset data that I downloaded. So this is just some path with some wildcards in it that expands out to all of those audio files. And so now I've told it what that configuration is. Now I'll run the setup cell, and now this is going to actually load that model from TensorFlow Hub, and also enumerate all of the files that we're going to embed. So in this case, it found 1,612 source infos, which are basically those WAV files that we specified. And then finally, we can run the embedding. So we'll do that. And this will take about, I think it took about 2 minutes when I ran it last night. And yeah, so that's chomping along and getting a couple extraneous errors that we don't really care, warnings from the new TensorFlow version that we don't really care about. And as this clips up to speed, yeah, so we're getting about 40 iterations per second. Each of these iterations is one of those input files. And for this particular problem, they have 1 minute audio files. So we're getting somewhere between 20 and 40 minutes per second using say 3090 GPU. The embedding model we're using, it's an EfficientNet B1. So like sort of the second smallest thing you could call a quasi-modern convolutional network. Many of you in the question time are going to ask why we aren't using transformers, and we'll come back to that. And yeah, and in about 50 seconds, we'll sort of get to the bottom of this. So that'll finish up shortly. And what I'm going to do now is swap over to another notebook. Now, yeah, so let's come over here. And now I'm just going to open another notebook. Now we've split these into 3. We looked at sort of putting them together into one and did that briefly, but actually the field biologists liked it a lot more than when they were split up into 3. So this embedding step, we really do it once, and then we work with those embeddings repeatedly. So, and the embedding step, especially when we get up, this is like 30 hours of audio, and we might have thousands of hours of audio that we're working with. So that embedding step, it can take a long time to run. So typically somebody who's a project lead or sort of tech lead will go through the pain of getting that to actually run end to end on thousands or tens of thousands of hours of audio, and then tell people when things are ready. Now, at that point, we get to this Agile modeling notebook. And so we'll connect to our local runtime again and get that set. And now, hey, it's another day. It's another notebook not authored by Google. And so we'll set up our stuff. I'm working with the file system, not some online things. I'm going to remind it what my working directory was. And just to show off a little bit, maybe at this point, I'll bring in the separation model so we can play with one of those, and we'll get those loaded up too. And so now we'll load this up. And one of the tricks here is, we actually told it what our working directory, where that was. All of the embeddings are sitting there that we just computed. Oh yeah, and hey, it finished in 2 minutes, just like I said it would. And it successfully processed all of those source infos and didn't fail at all, which is great. So now we've loaded up a separation model and the classification model, or the embedding model we're working with. And now we can start playing with some audio. So, we're going to start that process with search, right? So we want to search for something. And we've got a few different ways that you can, find a query. So one is we can start with IDs from this website called Xenocanto that hosts a whole lot of bird sound. And they've got hundreds of thousands of examples that people have uploaded at this point. So we can put in a random number and get a random bird sound. Hey, there we go. Beautiful sounds. And then we can tell it, we're working with 5-second embeddings with our model. And so we can also tell it, what part of this sound do we want to start with and pull out 5 seconds. So sort of defaults to the first 5 seconds, but we can move that around. So let's take an example from our actual data set that we're working with. So let's say we looked around just a little bit and found this cool file, which, in the first 5 seconds here, we've actually got 4 different frogs vocalizing. So this phi-saw, I am again just like really, really sad that we don't have audio here, because this is really quite an amazing sound. It sounds kind of like it's almost like a crying cat. It's like, I'll go grab my cat and torture it, and we can see if we can get something similar to actually come through the microphone. So this is the phi-saw, which is the that waxy tree frog, and then this sort of chirpy click up here, that's the lesser tree frog. And these other two, I don't remember what they are at the top of my head, and it's not a data set I helped build. So we can apply separation to that audio and see what we get. And in this case, it does kind of an okay job. So we do get, one of those Dinmin or tree frog vocalizations isolated. We do still have sort of a mixture for the waxy tree frog and whatever this other clicky one is. And we get that third one sort of on its own. Oh, and there's one more of the Dinmin sort of on its own as well. And hey, there's all of the insects that are sitting at the top of the track. And then here's some background noise. So the separation algorithm, it has some number of channels it's working with. And so it sort of pulls things apart as best as it can. But it's sort of trading off, what are actually the pieces I can pull apart here. And once it sort of runs out of channels to put things into, then it might end up putting things in the same channels. But, that's still better than we were. I looked around a little bit last night. I think if I look at, say, like 32 seconds, I'll get something that is, so then this one, we've got just our 2 target species, the Dinmin and that waxy tree frog. And somewhat unsurprisingly, this much easier example, it's going to do a much better job pulling this apart. So there's a really clear separation of our 2 Dinmins. This one, it's interesting. It's getting that, There's some normalization stuff going on here too, but this is actually quite a good example here. So let's look for this guy first. So that's channel 4. And I happen to know that the short code for this is phi-saw, so we're going to call it that. And we're going to work with channel 4 from that separated audio. If we aren't using a separator, we can just set query channel to 0, and we'll just use whatever the raw embedding was from the raw audio. So we'll select that. There's what we've embedded, just sort of as a last sanity check before we run the search. And now we're going to run the search. All right, and now we're going to wait for 20 minutes. Oh, it's done. Okay, cool. Great. So we've got some results. So because we've pre-computed those embeddings, we're able to compare very efficiently to them. And even with just brute force search, we're able to run over apparently 4,400 minutes worth of audio per second doing that comparison. So we're able to get results very, very efficiently. And these red points up here, so we've had a target score of none. So we're just going to try to get the best results here. And that's the stuff sitting up top. And then we can start taking a look at those results. And indeed, what we see is a whole lot of waxy tree frog. And we've got some buttons that we can start clicking. So, hey, yes, yes, yes. Yeah, so we've got some there. And yes, yeah, this is what we would call a chorus. So lots of them going at the same time. And, there's a whole bunch of these. I'm going to bore you for a moment and actually click on a few so that we've got like a little bit of data to work with. And okay, cool. So then we can write those examples out. So we've written 12 examples for our labeled data. And now we can come back and we can, let's change our focus and look at that lesser tree frog. So that was on channel 2. And that one short code is Dinman. And we're looking at query channel 2. So, hey, there's our embedding. And okay, now we're going to go for 20 minutes. Oh, no, it's 20 faster. Okay, cool. So we're good again. And we can look at those samples that we got back from that. And indeed, we're getting a lot of these tree frog examples again. So that's pretty good. And we can go through and click on a bunch of stuff and throw those in our training data set opportunistically. That's good. That's good. And at this point, yeah, sorry, if we were doing this for real and I hadn't done it last night already and sort of knew that all of these results are good, you would actually want to look at the spectrogram before clicking the button. But we're just going to run through quickly and get some positives in there. And yeah, and so a couple things to notice here. We're like getting good results even though they're in different parts of the frame. We're getting good results even though there's like, sometimes just one or two and sometimes many. So, we're actually getting some pretty good semantic embedding here. So there's 16 of those. And then we're going to take those training examples that we just picked out. And this sort of, we're working on overhauling some of this, but right now what it does is it computes a new embedding for those, which makes it easier to compare quality of different models. But on a day-to-day basis, it's not really necessary. And then we can train our small models. So what we've got it set up to do here is take 90% of those labeled examples from each class, use those for training, and then hold out the remaining 10% and use those for evaluation. And then it randomly does that process 3 times. So selecting 90% for train, 10% for evaluation. And then we get some model score averaged over these 3 runs. Turns out these are pretty easy classes. And so we've actually got a really high ROCAUC. It's really obvious what we're looking at here. Now, if I want to do some evaluation, I can say, hey, let's look at some phi-saw. And let's start with none. So that'll give us our top results and see what we get. So now we've got our logit distribution here, which looks like this sort of funny bell curve with some broad wings. And we can look at what our top results are. And indeed, our top results are all phi-saw. Now, when I look at this logit distribution, this kind of doesn't look great, right? So I've got this huge cluster around 0. And if you think about what the classifier is telling us, that logit 0 is this moment of maximum uncertainty. So that corresponds to the model saying, hey, 50% chance of being a positive or a negative. And so it's actually helpful to go look at what are the examples around that point of maximum uncertainty. So what we're going to do is we're going to change our target logit to 0.0 and see what we get. And we'll look at the results from that. And what we have are a lot of things that are not phi-saw. So when we trained this model, all it was looking at were the phi-saw and the Dinman examples. But it turns out there's a whole lot of other stuff in the data set and a whole lot of other embeddings out there. So what we can do is now we can go through and just sort of say, hey, yeah, like these are neither Denman nor Fisaw, when that's the case, or if we do find one, then we can mark those as such. And what this ends up doing is basically giving us some further support in the embedding space for kind of the negative class for these two classes that we're looking at today. So we'll go through and click a bunch of those, and then we can add those to our training set. Oh, looks like maybe I misclicked or something. Well, we'll have a mislabeled example, label noise, it's real. Then we'll reload our data with these new examples that we just added, and we'll train up the classifier again. Waiter, so much slower than search, right? Like, why would you even bother? And then we've got our new classifier, and now I can run this on Fisaw again, and I should see a really different distribution of logits. So we've sort of pushed a lot of that zero weight to the left, or a lot of that negative weight to the left. And now my uncertain examples should look a little bit more ambiguous. And indeed, they are. And it looks like they largely contain our target class, but sometimes at lower signal to noise ratio, and sometimes with just a lot of other stuff going on. So if we were to listen to this, this would actually probably be quite faint and with a lot of very loud competing sound here. But we can label them and say like, oh yeah, that one looks like it doesn't have one. So we can call that unknown, and so on and so forth. And then we can repeat this process for our other class and just get some further examples in there. Why do I keep getting one Fisaw? Interesting. Okay, cool. So that's sort of how that works. Oh yeah, because we actually found one. Yeah. Great. So that's kind of the basics. We can sort of iterate on that until we're sort of happy with the performance of these two classifiers. And then once we're happy, we can actually write that custom classifier out and save that for later. We can also run the classifier over the entire data set and see what we get, and put those in a CSV file for later analysis in R. And we've also got a third notebook, analysis, which we can use to basically look at like what's the actual call density, or what's the actual signal density within this data set with some confidence bounds. That's really great for all sorts of downstream analysis. And it looks like I'm a bit short on time, and you guys probably have some questions. So maybe I'll just stop there.
Awesome. Thanks so much, Tom. Yeah, there are quite a few questions here. Some from the slides and some from the actual tutorial. First one is, how do you account for the three-dimensional nature of aquatic environments in soundscape analysis instead of the more two-dimensional approach often used in terrestrial studies?
Yeah, so directionality is sort of the question here. So it's there's a couple of ways I can come at this question. So for terrestrial, mostly we're dealing with monophonic recording, so it's not even two-dimensional. And the reason for that is really, if you've got some extra money and you've got a choice between better microphones and more microphones. And typically people choose more microphones. So spread it out over a bigger landscape, get more data collection points, and then you end up with sort of a wider net that you're casting over. That wider net gives you a two-dimensional array sort of at the study level, right? And then you can use that for your downstream ecoacoustic analysis. So like, how is the ecosystem, or what's the response to different sorts of variables like elevation? There's your third dimension. Underwater, it's, actual hydrophone arrays are still very rare, and things are often attached to like a buoy or a boat, and are typically, like 100 or 1,000 times more expensive than terrestrial deployments. So typically you've got a particular kind of animal that you're trying to monitor because it is so expensive. What is it that we are spending all of this money looking for? And you actually know a lot about, what depths you should be looking at and where you should be looking in the first place. So yeah, so it's not really a problem that I think I've heard come up too much in practice. There are some really interesting studies, though, on like triangulation and some really interesting differences in triangulating in air versus underwater. But I'll defer that for another day.
Gotcha. Cool. Thank you. The next one, as you said, would come up. Why a CNN over a vision transformer?
Yeah. So if we go back to the it's actually kind of funny. One of the most constant things we've seen is the things that people think should work based on the common wisdom in the machine learning community at the moment seems not to be doing a great job. So like this audio MAE it's basically a vision transformer on spectrograms, doing a self-supervision task trained on massive amounts of YouTube data. So a lot more data in terms of volume than the bird training data that we're using for the classifier. And we see it's underperforming. When we train transformers and self-supervised methods on soundscape data, we've often found that they tend to really overfit whatever the training distribution is and fall on their face when we go to the actual transfer learning task relative to the features that we get out of our convolutional networks. So what we really care about is transferability and generalization of features. And we're getting much better results right now with these global bird classifiers.
Sweet. Thank you.
We're still working on it,
though. Cool. Yeah. So the next question is, how many hours of data should be collected for good results? What are the memory requirements? And if a model was developed for detecting noise pollution, would this approach work?
Yeah. So that's a lot of questions. So one of the nice things with the bird classifiers, it is actually really cheap in terms of memory, right? Like you can run these on a phone very easily. So memory is not a problem with our current breed of model. Data set size. So there's like the ecology question, the machine learning question, right? So like we've just seen, we can get a really good frog classifier for these target classes with really just a handful of examples. So, do you have enough audio to find a few examples of what you're looking for? If yes, there's a good chance we can train up a good enough classifier. And then for the ecology question, it's an entirely different question. Like, what is your study design for sort of covering this landscape and getting out the information that you need to figure out whether restoration is working and, and all of this stuff. That's at least 2 out of 3. I forget what the third one was.
Yeah. And the third one was, if a model was developed for detecting noise pollution, would this approach work?
Yeah. So it would be really interesting to try. So on some level, noise is not birds. So it might be that the, characteristics of background noise might be lost by the time we get to species classification, but you can try it and see what happens. You could also, if the bird model doesn't work, I mentioned before, you can actually pick up any embedding model you want. And we've got some things that are trained on AudioSet. And AudioSet has a lot of classes like, is this a passing car or is it an air conditioner? So it might be that those sorts of models are much better trained already to address these, like what are the characteristics of the background noise kinds of problems.
Gotcha. All right, so I think we have time for one more question. So when you were first labeling the data, there weren't any negative examples labeled. Do you only need positive examples for this?
So we actually went back in the second round and got a bunch of negative examples. And that was what helped shift the distribution and reduce the uncertainty around those things that weren't already there. What we're really leveraging is that for these bioacoustic cases, the embedding space is sort of pre-clustered, right? So if you've got a funny little cluster of stuff, you need to find the hyperplane that separates it. But when you put that hyperplane down, it sort of tells you about this cluster versus this cluster, but there might be other noise examples that you don't care about that might be on one side or the other. And usually just like one round of going and looking at some of the ambiguous examples is going to give you enough unknowns that you can do a good job.
Gotcha. Awesome. Thanks so much for your time, Tom, and for answering these questions afterwards. Yeah, any last words from you?
Um, let's see. Yeah, I mean, I think just seeing a couple of the other questions here. So like in terms of signal processing, like, hey, this is signal processing. Like there is signal processing in here. But it's also much more robust and much more efficient, I think, than if you look at older approaches, it's sort of like, let's develop a click detector, and that click detector is going to misfire on all sorts of stuff. So what we're getting here is really a much more robust embedding. Why are we getting a robust embedding? Because there is an incredible amount of diversity of birds, ranging from 300 species of doves. So, even thinking about distinguishing those in sort of the lower registers, you have to develop good features for that. Likewise, all of that complexity that we saw at the beginning around songbirds, you need a feature set that helps you distinguish all of that complexity. So we get these really, really robust embeddings, which we're then able to leverage into this very efficient workflow. And I think the last piece I'll throw in is, model quality is cool and all, but I think maybe the most important thing that we're thinking about here is how do we actually save time for people working on the land in conservation? So, sometimes it's nice to make the quantifiable, model quality metrics go up, but what I really love to see is the amount of time that people have to spend futzing around with computers when they could be going and doing the actual restoration work go down. All right. Thanks a lot.
Thank you, Tom. Thanks everybody for joining today. Tomorrow there will be a lecture on AI for climate science, so we hope you all will join us there. Have a great rest of your day. Bye bye.
Cheers.